Hey guys, so this is another interactive lesson that you do on your own at your own pace. You'll need your headphones for this and your note-taking guide. This is what your note-taking guide looks like. So we're going to be doing 6 sections, and the 6 sections here are following the same order. You're going to find the definition, what is involved, how this advancement can be used. Are there any ethical issues that may arise? Ethical issues are moral issues. Is this right or wrong to do? And you'll also be comparing any positives or negatives. So as you go through the topic, just go down the column to fill out the notes. So this unit, as you know, we're talking about biotechnology. Biotechnology is what scientists use organisms and their products, so animals and plants and the products that they make, to improve human health and food production. So manipulating the DNA to make a bigger or better product that will serve our needs. So take a look at this picture. Is this picture true or exaggerated? Scientists have been manipulating our food for many years. Take, for example, the super tomato. Super tomato has been manipulated. The scientists have changed its DNA so that the tomato would be bigger or redder and last longer on the shelves so it could be transported. The picture is exaggerated to make a point because scientists don't inject every single tomato, but they have been manipulating the tomato DNA in the plant, and the plant will produce bigger, better tomatoes. So, in a nutshell, biotechnology is manipulating DNA and understanding how DNA and genes are passed down from parent to children, and using that information to make bigger and better organisms, or use these organisms to produce products that are useful to humans. So let's go on to the first section, selective breeding. Selective breeding, you may have heard this when referring to dogs. Dogs have been selectively bred for hundreds of years because dogs come from wolves, and we decided which traits would be passed down from parent to children. So selective breeding, the definition is that desired characteristics are selected to reproduce. So if you look at the picture, how are that little dog and that big dog related? They're both dogs, and they both come from the family of wolves. They're the same species, but they are very different because they have been selectively bred. So that means that the tall dog's parents were tall, and the little dog's parents were little for many, many generations. So if you take a look here at the genealogy or the family tree of the dogs, you can see that all dogs come from a common ancestor, that is the wolf. And by deciding which male and female you're going to reproduce and make babies, you decide the outcome of the babies. So say, for example, you want only white puppies, you're going to take a white female and a white male, and you're going to reproduce them, and you're going to have a litter of puppies. Now, if any puppy comes out with a different color, you put them aside, and you only reproduce the white puppies from that litter. So the next generation of kids will all be white, and that's how you selectively breed animals. Now, if you buy a certain breed of dog, they're called purebreds, or a pure breed that you know that they've been those characteristics for many, many generations. Now, plants have also been selectively bred. If you look at the corn here. Corn was never the size it is today. Corn used to be very tiny, as you can see over here on the left. And by deciding which corn to reproduce for the next year to make seeds, we selected the corn with the bigger husks or the bigger kernels and reproduced those year after year until our corn became larger. Same thing with carrots. In nature, carrots are... So as you see, even the carrots come in different varieties. Carrots come in red, orange, yellow, even white. But we've selected only orange carrots to reproduce for the supermarkets. So only at the supermarkets you only see carrots, orange carrots. Here's some example of animals. Cows are selectively bred. Female cows that produce lots of milk or good quality milk, those are the cows that are going to be reproduced so that their children can produce high volumes of milk or high quality milk. Same things with bulls. Strong bulls are used to reproduce so that their good genes are passed down to their children. So we select which characteristics are going to get passed down. And again, with the dogs are selectively bred so that their children come out the way we want them to look. Like for example, a wiener dog. The wiener dogs are not natural in nature. They don't come out by chance. We selected those traits to be present in the generations. So what is involved in selective breeding? Sometimes hybridization is involved. What's hybridization? You might have heard this word when you talk about a hybrid car. What's a hybrid car? A car that runs on both electric and gasoline, two different methods. So when you talk about a hybrid, you're joining two things together that are very different. So hybridization is you're breeding two different individuals for a better offspring. You're breeding two different species so that the children come out better than the parents. An example of hybridization is when you reproduce a horse with a donkey. What do you get? You get a mule. Now a mule is neither a horse nor a donkey, or it's not even a species on its own. It's a hybrid of two different species. Same thing as when you reproduce a lion and a tiger. A lion is one species and a tiger is one species. The liger is the hybrid of those two species. Here are some websites that if you wanted to check out to learn more about selective breeding, you can click on the link and see if it takes you to the supplementary information. And now we'll move on to the second topic. The second topic is cloning. What is cloning? Cloning is when you take an individual and you create identical individuals from just one cell. All you need is one cell. So we can do this with animals, and they're trying to do it with humans, but as you'll see in a bit, what ethical issues arise when that happens. Let's talk a little bit about what is involved with cloning. So if you look at this picture here, you take a cell and you fuse it with an egg, okay? And then you take that egg and you put it in a foster mother, and that egg grows into a new individual, identical to the donor of the cell. So this is the same picture zoomed in. As you see here, they take the black sheep, and the black sheep donates an egg. Okay, they remove the nucleus, okay, from that egg. So they remove all the black sheep's DNA from the egg, and they take the white sheep, the white-faced sheep, and they take one cell from him. They fuse that cell with the egg. Now, inside the egg has not the DNA from the black sheep, but actually the DNA from the white sheep. So when that embryo starts to grow, it's going to have the DNA of the white sheep. So they take a surrogate mother to hold on to that egg and grow for a couple of months, and then that sheep is born like a normal sheep, but it's identical to the white-shaped sheep. That's cloning. Why would we want to clone animals? Here's an example. A good application of cloning is to save endangered species. For example, like pandas or certain tigers that are endangered, we can use cloning to reproduce those animals without the fear of them dying. What are some negatives that are associated with cloning? Well, there are ethical issues involved with cloning. Can we clone humans? Should we clone humans? Do these clones have the same rights as humans born from an egg and a sperm? Or do these clones have no rights and we can do whatever we want with them? Remember, they're born the way me and you are born, but they share your DNA. So where do we keep them? Do we keep them in the hospital? Do we let them go to school? Do we educate them? Do we give them language? Do we teach them how to read, write, and speak? Do we allow them to go to high school? So all these questions come to play. Should we clone humans? You'll probably remember a great example of cloning in a movie. In Jurassic Park, scientists were able to take DNA that was DNA of dinosaurs that was in the blood of the mosquito. So the mosquito sucked the blood of a dinosaur and then got stuck in the sap, if you remember the film. So scientists were able to take the blood that was still in the mosquito and put it into eggs, reptile eggs. So they were able to clone dinosaurs. And we all know the bad things that happened in that movie. Here's some supplementary information with cloning if you want to learn a little more about it. The third section is genetic engineering. What is genetic engineering? Well, it's when you take pieces of DNA from one animal and you put it into the DNA of another animal. Or you take the pieces of DNA from one plant and you put it into another plant. Okay? Joining this DNA will now change the characteristics of that animal or plant that you're putting it into. So what is involved? You're taking recombinant DNA, okay, like recombination, okay, recombinant DNA. You're joining DNA from different organisms. So you're taking the DNA from one animal and you join it with the DNA of another animal. Okay, so if you look here, this is just a little cartoon. This is the original DNA of, let's say, a bacteria. We are forcing that bacteria to take on this green DNA, which belongs to another organism, and we insert it in the DNA of that organism. So now that bacteria has foreign DNA in it, and it is going to act and look a different way. Here are some examples. In college, bacteria are great organisms to experiment on because they reproduce very fast and their DNA is actually very simple. But in college, we took some E. coli bacteria and we inserted glow-in-the-dark DNA, the DNA that allows the animal or plant to glow in the dark. So we inserted it and we made those bacteria glow in the dark. It was actually a pretty cool experiment, and it was nice to see how we can manipulate the DNA of an organism. In plants, this is being done very often. in tomatoes. Tomatoes are getting injected with DNA from other plants, as well as strawberries. Strawberries carry extra chromosomes and extra DNA from other plants to make them grow bigger, because in nature strawberries are actually very small, but as you can see here and in the stores, strawberries are actually very large, and that's because we've added extra DNA to the strawberry plant. Here's some extra activities if you wanted to look into genetic engineering a little more. Let's move on to our fourth. Our fourth section is transgenic plants and animals. Once you change the DNA of a plant and animal, it becomes transgenic. That means now they carry the genes of some other plant and animal within them. So as we can see here, corn has been genetically modified. That means corn now has the DNA from other plants and animals. Scientists put it in there because if you see here on the picture on the right, corn in nature should look like that. Okay, very thin, very fragile, but we see how corn was genetically modified over the past 100 years to now be bigger, larger, and much more producing so that they can sell it. Here's another example of transgenic animals. Fish have been genetically modified, so they change their DNA so that they can grow faster and larger, and they can be sold for more money. So this is genetically modified fish, and this is a normal fish that was caught in the ocean, and these are the larger fish that were grown to be sold at the supermarkets. Same thing with chickens. Chickens are highly genetically modified because there's big business in making chickens. They have changed the chicken's DNA to be born without feathers. So those chickens don't have feathers, so it's easier for them to kill them, take their skin off, and sell those chickens at the supermarket. They also made them grow faster and grow bigger and fatter and have particularly large breasts so that they can sell the chicken breast at the supermarket. And if you notice, these chickens are not active because they are so heavy. They end up dying very quickly, and so it leads to other health concerns. So this is of interest to you because you should know where your food comes from and how it's being taken care of, so that when you go to the supermarket, if you see that your food has been genetically modified, or that it comes from GMO, genetically modified organisms, you know that their DNA was played with. So you may be cautious or may want to learn a little bit more about where your food comes from, or if you decide to eat it or not. Another way we use genetic engineering is to use animals to our benefit. For example, many people have to take insulin shots because of diabetes. And now insulin is a protein, I believe, that is produced only by humans. So what we did is we took the DNA from humans that produce insulin, and we inserted it into the cow's DNA. And so now the cows with the human DNA inside them produce this insulin, and all they have to do is take it out of the cow's milk and then put it into a syringe. So now we're basically using the cows to create medicine for us. The fifth section here is DNA fingerprinting. If you look at your hands, you see your fingerprints. Everyone's fingerprints are unique. Nobody has the same fingerprints, not even twins. So that goes the same for your DNA. Nobody shares your DNA. Your DNA is unique to you. So if you commit a crime, they're able to pinpoint you if you leave your DNA behind. Or also, if you want to find out who your baby daddy is or who your mother is, using DNA sampling, we can find out those answers. And this is what a DNA fingerprint analysis looks like. There's these lines here, and I'll explain in a bit what these lines do and how they can determine whether your DNA belongs to you or somebody else. So, what is involved in DNA fingerprinting? This is what happens. You take your DNA sample and we put it in this little machine here. It's like Jello, and we insert the DNA samples into this Jello, and we attach an electric current to it. The electric current will pull the DNA down the Jello, and the DNA will slowly move down this gel, and how fast it moves depends on each person. So each person is going to have a different design when it comes to their DNA. So each person, this is like a picture or a snapshot of your DNA. So how can we use this information? Paternity tests, finding out who's the baby daddy. So let's do this example, and then I'll show you how the gel electrophoresis works and the DNA fingerprinting works. So Sammy from the Jersey Shore is pregnant, and she believes that Ronnie is the father of her baby. Ronnie isn't so sure if it's his baby because Sammy has been with both Mike, the situation, and Ronnie. Scientists took several DNA samples and performed a gel electrophoresis experiment. So they take samples from the baby, the mother, and the two guys who are suspect. So Mike is male 1 and Ronnie is male 2. Let's take a look at the results. So scientists took samples of their DNA and performed the gel electrophoresis experiment. This is the results of the mother. This is the results of the child. And if you notice, about half of the lines are similar to the mom. Why half? Because half of the DNA comes from mom and half of the dad. Which one of these resembles most the father? Male 1 or male 2? Let's look at similarities. If we see here, this child is similar to male 1 here. This line, this line, this line, this line. That line is similar, this line is similar, and that line is similar. So it's very obvious that half of the DNA comes from male 1 or Mike. So who is the baby daddy? In this case, the baby daddy is Mike. And as we can see, all the purple highlights are the similarities between the mom and the child and between the child and the dad. Okay, so good. Crime scene investigation is another way that we can use DNA to pinpoint the criminal. For example, let's do this scenario. If a car was stolen, a stolen car was found on the side of the road with an empty bottle of Sprite. Detectives were able to collect enough DNA from the saliva left on top of that bottle to create a DNA fingerprint. So the criminal leaves behind his bottle of Sprite in the car he just stole. The scientists take his DNA from that Sprite bottle because he backwashed. And let's see, there are a couple of victims here, the victim and the suspect, and the DNA found at the crime scene. So the scientists do the DNA fingerprinting, okay, and this is the DNA fingerprint of the crime scene. This is of the victim, so we can see that the victim of the car, the owner of the car, has nothing to do with the actual DNA. There's no similarities. Suspect number one is a identical match. Look here. The crime scene DNA and the suspect DNA are an identical match. So suspect number one is the guy who stole the car. And we see here, suspect number two has his own identical DNA, but it doesn't match the DNA of the crime scene. Here are some resources to learn a little bit more about DNA fingerprinting. And then the last section here is the Human Genome Project. This was a project started, I believe, 20 or 30 years ago. Scientists wanted to identify all the genes of the human body and all the DNA sequences. If you can imagine how large this project is going to be, because there have been identified more than 25,000 genes, and those genes are the code for all the proteins in our body. What is involved? We have 3 million DNA-based sequences. What does that mean? Remember when we studied DNA? And DNA has a special code in 4 letters, A, T, G, and C. Well, those codes determine who we are as a person. And each one of us have 3 million of these letters lined up in each of our cells. So, and we're each individual, so that means that we have different combinations of these letters, A, T, G, and C, 3 million of them. So you can imagine how hard this project and how long it would take. So why would we want to know all this information about our DNA? Well, how can we use it? We can use this information because if we understand our DNA better and know where everything is in our DNA, then we may have hope to detect and treat diseases, because most of the diseases are passed down from parent to children. What if we can remove the gene that controls cancer and we can cure cancer? Or what if we can determine the gene that causes Alzheimer's or Parkinson's disease or diabetes or strokes, and remove that DNA and replace it with healthy DNA? Then we can probably lead more healthier lives, or who knows, maybe even live forever. So, of course, all these technologies that we have come with a cost. Are there ethical issues with the Human Genome Project? Yes. It may seem all good and dandy if we understand our DNA better, but what risks do we take? Well, the positives are that we can identify and treat diseases, as we spoke about before. But the negative is that if we map out all our DNA, there's a negative to it. We may be in a situation where we can lose our human rights and we can lose our privacy. Why do I say that? Because, say for example, when you're born, they take your DNA and they map out all your DNA and they put it in a book, maybe a passport. And now with your identification for the rest of your life is going to be determined by what your DNA says. So if your DNA says that you're going to have a heart condition when you get older, maybe you will be prohibited from joining any sports in high school or college. Or what if you are prone to a certain disease and your future wife or husband finds out? Then they can divorce you or never marry you in the first place. So you lose this privacy and you lose your human rights, because now it's public knowledge that everybody knows what your DNA reveals. Another example would be alcoholism. They're starting to find out that there's a gene for alcoholism. So alcoholics have this gene, it's in their DNA. So say you had this gene for alcoholism, you would probably be prohibited from buying liquor. So it limits the freedoms we have as humans. More knowledge means more responsibility. This brings us to the end of the presentation. Now go back if you missed any sections of this presentation and fill in the sections of the matrix accordingly. Thanks for listening.